Methods for purifying and studying proteins. Protein purification and analysis are fundamental techniques in biochemistry and molecular biology. These methods allow researchers to isolate specific proteins from complex mixtures, study their structure and function, and develop new drugs and therapies. Protein purification methods. Several techniques are used to purify proteins. Cell lysis, physical methods, sonication, French press, or bead milling disrupt cell membranes. Chemical methods, detergents or chiotropic agents solubilize proteins. Enzymatic methods, lysozyme or other enzymes break down cell walls. Centrifugation, differential centrifugation, separates components based on size and density. Density gradient centrifugation, separates components based on buoyant density. Chromatography, size exclusion chromatography, separates proteins based on size. Ion exchange chromatography, separates proteins based on charge. Affinity chromatography, separates proteins based on specific binding to a ligand. Hydrophobic interaction chromatography, separates proteins based on hydrophobicity. Electrophoresis, SDS page, separates proteins based on molecular weight. Isoelectric focusing, IEF, separates proteins based on their isoelectric point, pi. 2D gel electrophoresis, combines SDS page and IEF for high-resolution separation. Protein analysis methods. Once purified, proteins can be analyzed using various techniques. Mass spectrometry determines the molecular weight and amino acid sequence of proteins. Spectroscopy, UV-vis spectroscopy, measures protein concentration and structural changes. Fluorescence spectroscopy detects intrinsic or extrinsic fluorescent probes to study protein conformation and interactions. Circular dichroism (CD) spectroscopy measures secondary structure of proteins. X-ray crystallography determines the 3D structure of proteins at atomic resolution. Nuclear magnetic resonance (NMR) spectroscopy provides information about protein structure and dynamics in solution. Enzyme-linked immunosorbent assay (ELISA) detects and quantifies specific proteins using antibodies. Western blotting identifies specific proteins in a mixture using antibodies.